Studying the tumor microenvironment is difficult due to its complexity. We've developed microenvironment microarrays consisting of simple combinatorial microenvironments that allow the identification of major drivers of tumor cell phenotypes. The major advantage of the MEMA platform is that because the microenvironment conditions are all defined, it is straightforward to identify microenvironment factors that drive phenotypes of interest.
The MEMA platform is widely applicable in other non-cancer systems, including primary cell strains and stem cells.
There are a number of engineering tricks and steps that we have developed to speed the wet bench work and simplify the protocol.
To begin, use a touchpin printer to print extracellular matrix print mixture in fiducial spots into 8-well plates. Use 350 micrometer diameter pins arranged in a 4x7 printhead configuration to print the extracellular matrix for the microenvironment microarrays. The collagen block is printed onto MEMAs as a grid. The PBS-filled wells provide extra humidity to prevent drying out of the extracellular matrix print mixture source plate. Print the arrays in the 8-well plates as 20 columns by 35 rows for a total of 700 spots. After printing, store plates in a desiccator for a minimum of 3 days prior to use. First, design a 96-well plate layout with spacing that allows for the use of a multi-channel pipette with 4-spaced tips so that the treatment of many MEMA plates can be done at once. Then, retrieve the ligands from the freezer and thaw on ice in a laminar flow hood. Briefly flick and spin down.